all the samples together, but maintaining what those IDs are across those samples for the cells in question. So just kind of pulling up the cell-Loupe browser. So this is the aggregate of all the samples, just shown in the cell-Loupe browser because it's easy to show initially. And this is on the box folder that I shared with Banga already. And I can share it with the rest of everybody too. So here it does just basic clustering. It's a graph-based algorithm. There's a couple of different things. You can either do it as a graph-based clustering or a k-means clustering. Came up with 17 different clusters, things like that. The main important thing, though, is you can use this easily to check for any type of like batching effects and things of that nature. So if we go through and assign the library IDs to each of these cell points, we start seeing kind of a separation here with the majority of the 3' samples being down here.
Right.
And then the 5' samples are the ones that are basically up here. So that was a little concerning. I wasn't too sure if we can do the aggregate across these different chemistry sets or not. It doesn't specifically say whether you can or can't on the website. It just says that you can't use the first version of the 3' chemistry with either of the other ones. So that kind of implies that you should be able to aggregate across it, but doesn't explicitly say that you should be able to. So I thought maybe, we were seeing the split here because of that. It could have been because of that chemistry. So I also did this just as separate, two different aggregates, one for just the 3' samples and one for just the 5' samples.
Oh, you did do that? Just the 3' and 5? Oh,
yeah, I ran
I
those today. It runs pretty quick, so it's not hard to run those. So this is for the 3' samples. And again, it does the clustering and everything. So if we pull up the library ID, we do still see some of the separation here. So specifically, sample 135 is very separated. So that's kind of the ones that are flashing on and off there are 135. Those are a bit more separated from the other two samples.
Yeah, 135 is me.
Your healthy control, right?
Yeah, right.
They're not very healthy.
I'm not too healthy. My B cells are a little screwed up.
Okay. And then,
it's not like I'm uninterested, because I do have a little problem there, and, it might be interesting to figure out what it is, but, I'd like to look at 135. I could write a case report on 135. I wouldn't mind doing that, because I know the history of it, too. But anyway, that's something else. If you want to write a case report, there is one.
So there is a bit more overlap between 128 and 132, but you still see a little bit of separation here. And then we can look at the 5' one as well. Now, I'm not too sure kind of why we were seeing things. So this is the separation for the 5' chemistry, with this blue one being the healthy control, and these orange and green ones being the SLE ones.
That's quite interesting. And one thing we're going to do, Chris, is we're probably, 3' was what was available when we started, and now we're probably everything will be 5' and hopefully comparable from now on out.
Yeah. Yeah, that's good to keep it all up, the same chemistry and everything. The one thing, though, is we're still seeing quite a bit of separation here. So that's 166, whereas these ones are all 165.
That's quite
it
interesting.
kind of can imply a little bit of batching effects. I guess for a comparison, we can look at this data real quick. Where was it? Yeah, this one. So just as a comparison, real quick, this is Nate's data, but he had 8 different samples, and you can kind of see that they're all spread out a bit, but there's a separation based on treatment groups.
Yeah.
So, you know, these are one treatment group and these are a different treatment group. But within those groups, they overlap quite well. So if you take out all of these, that's, you know, the majority up here. And if you take out all, or if you just do these, you know, it's the majority down here.
I kind of like the way the bottom shows, you know, there's one sample down there, but, you know, the genes, I like to look at the bottom, and I can always see an outlying sample, but these are at least all, you know, highly expressed genes, sort of all on the left side. They're not all, you know, it's not perfect, but when you throw something strange in there, you know, it shows up real clear, you know, that all of a sudden there's...
So you're talking about this heat map?
Yeah, the heat map. It'd be nice, you know, one thing that would be really nice is to be able to, you know, present that heat map, the data in that heat map. And I think they went through it, the guy said, but somebody wasn't able to take the data that made that heat map and make another, you know, present that, because we can do another heat map with a, you know, that's more readable.
Yeah.
And, yeah, right.
We
you
can
know.
get the underlying data and do this in MATLAB if you want.
Yeah, because, you know, like, okay, that heat map, you know, you say, oh, that's quite interesting. Here's this one, here's this one. Are they the same or not? Especially the heat maps of the norm. You only got 2, but 3 yeah, there you go. Oh, that's even better. How'd you do that? Make it at least bigger square.
You can adjust it.
Where is that adjustment?
It's just a drag bar. Once you mouse over it.
You mouse.
You drag it.
Can you do that again? Oh, you're dragging it up. Oh,
You just go right on the edge
And
of
you
it.
just click and drag.
oh, well, that's what I'm talking about right there. That's good, because then you can see. And what'd be nice, again, you know, this is asking for a lot, but if the genes now could be displayed along the side on the I guess the x-axis in this case.
Yeah, as you mouse over them, it shows you what genes each column is. So the columns here are the genes.
Right.
And the rows are the clusters. So there's 13 clusters that are generated based off of the graph based clustering.
Is there a way to just put the genes, you know, down the x-axis, you know, along the horizontal there?
In terms of like labels?
Yeah.
Not specifically. It doesn't-
No, we'd
I
have
don't
to
think
export
there
it
is.
probably. And do it that
You
way.
export it. How do you export it?
Yeah, so you can export just the heat map image just using that little camera icon.
Oh, camera. Okay.
Yeah, but what about the
If
data?
you want lists of genes, you can either pull up, you know, there's a gene table icon here, and when you click on it, it'll list. So here, the rows are the genes, and the columns are the clusters. So you
Right.
can click on specific clusters if you want. So, like, say you're interested in cluster 1, this dark one, you can sort your p-values based off of that cluster and see what your top genes are.
Yeah.
So,
Can you export it?
yeah, so when you go to this little icon here, this exports the table.
Which icon? You know, it's okay.
There's a little arrow here.
Yeah, little arrow. Okay, export tape. Oh, I see. To CSV. Okay.
Yeah. And you can specify if you just want, say, the top 50 genes.
Right.
or,
Right. Oh, that's good.
I think, yeah, top 10, 20, 50, 100, or all of the genes.
Oh, that's quite, yeah.
And
Usually
it's
the top 50.
automatically hiding genes that have low UMI counts. So UMIs are the unique molecular identifiers, the barcodes that it puts on there. So
Right.
if there's a low count of those, like 1 or 2 probably, I think it's just 1 actually, greater than 1 transcription, so yeah. So it filters those out basically.
Could you do that just as an example? You know, because it sounds like you can do that pretty easily. How
Pardon?
can you go ahead and do that? Make that file and save it, and even email it to us?
Yeah,
You know, just
yeah, definitely.
'cause however you do it.
Like I said, I mean, you have this.
Top 100 is-
But I can also export these.
The top 100 is fine.
Well, they do the top 100 is based on p-value, right? So
Yeah,
so you could just do everything because it's only 1,000. It's not that.
Oh, okay.
It's not a huge amount. It's not really a huge
Well, we
amount.
could chop it off if we want.
Yeah,
I'm
then-
thinking of presenting something, and
maybe
like
we could
such as in the grant that I'm writing and due in a few weeks.
But yeah, so, I mean, this is just a nice interactive way to deal with it, kind of, and it helps show you, I don't know specifically what genes you guys are interested in. for
Actually,
these,
Hey, Chris, I have a question. Can you go back to the 3' example?
Yeah, this is the background.
Is that all 5', by the way? All 5' samples put together?
The one that we were just looking at was the 5'. These
And
are
there-
the 3'.
And there was like, I forget, but 12 clusters within, when you put all the 5' together, and the normal was on the one place, I forgot what it was, but, it's, even something as beginning as this is pretty nice.
Yeah.
Um,
Yeah, so that was the 5', 13 clusters, and yeah, you can kind of just colorize. Even if you wanted to look, if you have specific genes in mind, like this TCL1A, if you wanted to colorize these based off of that, you can do that as well. So you go to gene expression here, and then just type in TCL1A. I'm not sure what that is, but it shows us the clusters that have high expression of TCL1A. So
TCL1A
it might
actually
be
is,
hard to see, but the other
it's
cells are
actually interestingly, it's a T-cell leukemia, but it's in B cells. Somehow, how things get a bad name.
Well, are these? It was unclear to me if these are B cell. I thought these were just like PBMCs.
No, they're B. Well, TCL1A are their B cells. For example, I'm over there. I'm all B cells. I got a lot of B cells.
Yeah. I mean, it's just going to be like, I think for some of those cells, there might be like a minor contaminating like monocytes or T cells, but that's a very small fraction of the total cells.
So if the sorts are not precise for different individuals, that could also lead to the
Right.
person-person segmentation.
Oh yeah, and they're never 100%. I mean, they're pretty close. Like you see that big.
So you think,
Yeah.
Okay.
So like maybe one of these clusters. up
Yeah,
here.
That's
Yeah,
not
that one.
yeah.
I want to see. So which cluster is that one?
That's cluster 9.
Okay, can we look at the heat map for that and see?
It's different. I know that guy. He's a monocytes or something like that. He's not a B cell. Maybe he's
Well,
T
no,
cells.
it's just the shape of that cluster, I think, is going to be dominated just by a few genes, and I wanted to see if that was
Oh.
actually the case.
Oh right.
So
That's
if we
good.
show the
point.
heat map for that one. Because
Good.
when you're doing this for the RX data,
Oh, absolutely.
That's what-
Yeah, a few genes are dragging it out to the top there. I think.
Cluster 9 is.
Oh, here they are right
bottom
here.
row.
yeah,
Yeah. So you're right. These I guess these genes are
Yeah.
you know.
so they're just really highly
They're
expressed.
I see lysozyme and they're macrophage genes and none different from the. And I guess this one here. So I saw lysozyme and some other genes. What's this one down? Oh, you can't see. Cluster 11. There's another bunch of red. I
Yeah,
bet you.
there's-
What's this? What can you mouse over this one?
I don't know.
Okay, it's got the cluster 11, the red one.
Cluster 11 down here.
Because that's
Yeah.
probably T cells, I guess, because it's not lysozyme. This sign I can't tell. So CD7.
IL-32.
they are T cells.
Yeah. So you can tell those are
Yeah.
monocytes, macrophages, and these are T
Here
cells.
we see.
And where's cluster 11 on the plot, Chris?
It's probably down here.
Where is it?
Yeah.
It's some peripheral.
This
Yep.
section down here, yeah.
Okay.
Those are interesting. We don't want them to show in the final plot. We want to get rid of
Yeah.
that.
So different. If you don't have a really good sort, the different cell types is going to probably drive the most distinct clusters. Is what
Oh,
I'm
yeah.
seeing it this way. Probably.
And it's only a few genes, but we're not interested in them right now.
So
Is it, we keep asking this, Ray Chin asks, I ask, but if you get rid of those clusters,
yeah.
and then are those clusters influencing the,
So I mean,
The plot.
I mean, because I wouldn't know. Would you
Well, they
like,
definitely are, yeah.
leaving them out before
In
you
terms
go
of
with the-
this program, I'm not sure if there's a way to exclude
Well,
We're
but
throwing out
we
cells
asked-
based on the fact that
we
transcriptome
asked the
is
guy,
supposed to have T cells in
yeah,
macrophages.
this Monday or Tuesday, and he
Yeah,
says,
you're
what
right,
you can
it's-
do is
it's not-
export the data and then redo it. And as he said, but that doesn't make any sense. I said, no, it does make sense because you got macrophage and T cells that inadvertently got thrown in, and they're pooling,
How do you make-
distorting this plot. And it'd be good to get rid of them and then redo the plot and see if it's any different.
So you did
Tisney
sort,
plot, you know.
Just the output sort.
I don't know why that's
Yeah,
such a,
and
that why
so it
that's
was
not
like a
obvious that
negative
we want to do that, but.
In this case, so it was just like a negative
Yeah, so I mean, that might
purification.
be a good approach if that's kind of what's driving these separations.
Yeah, right.
And maybe, we don't necessarily have to re-import it back into this, but use like Syrah. Because a lot of this stuff you can also do in Syrah.
It'd be interesting to see. I mean, we're just going to turn them off. I mean, we're either going to turn them off, or we'll turn them, or we'll turn them off and reanalyze them. But we're not going to show them. We're not interested in them, and they're a contaminant. And everybody knows sorting is not 100%, and so these things are going to go away one way or the other. It'd be nice if they went away. Now that we know that we don't want them and they're contaminant, it'd also be nice if they went away for the analysis. We didn't even want them in the analysis. You know what I mean?
Oh, if you read.
But that's a small
Throw
thing.
the cells out and read-
Nobody seems to be able to. They say, no, you want
So that's
those.
true, we could
But
actually-
no, we don't want
Do Chris,
Yeah,
can you do that view where you highlight the particular genes and search for CD14?
CD14.
Is that the one you would just see the? That CD, oh. Yeah, that was one of the two cluster, that was also in cluster 9.
Yeah.
them.
Okay, now do, can you do CD3E?
CD3B is a
Yeah.
boy.
Epsilon.
E.
They're going to be up there in the T cell area. We know this. So we're not insulted on it, whatever, okay. The monocytes are there. Whatever they are, we don't want it. We know we don't want those. We don't want, the.
Yeah, how about CD19? That should be most of them.
Right, they're in the middle. Those are the ones we want. We still don't want them. Oh, I see the dot.
Oh, it takes a while to refresh.
Oh, okay, yeah. So we don't want C, so those are the ones we want.
So why do you think there's the concentration up in the upper left, but not so much in the middle?
Well, that's CD19. All the B's.
Because the cells that I'm interested in, they have higher levels of CD19.
Oh, so it's not just on or off. There's
Yeah.
some subset or some patient
It-
with elevated CD19.
But in some subset of their cells.
Okay.
Gene, as you know, gene expression is not the same as protein expression. And maybe it is, maybe it's protein expression, but there's a lot of B cells in there that just don't have that gene. It's one of those genes that is a very long life protein transcript. It sits on the surface, and the cell might turn it off. But it doesn't mean they're not B cells. The protein is there. If we sorted for CD19,
Yeah. I mean, there are like, I think like a combination of 3 that you could probably use to pull out,
yeah.
or at least 4 that you can use to pull out all the B cells.
Here's what I had a conversation about it with someone about I don't know 5 or 6 years ago, when this was just starting, and there was a paper that came out and says you cannot identify T B cells and macrophages with a marker, because everybody's saying, well, we don't have to sort them, we'll gate them out later. But the thing is that, and then a paper came out that won't work. And I said, oh, really? That's strange. But then they said, well, genes are turned off even though the surface marker's there. But, so that's it. I mean, it's not hard to understand. It's just that the genes are different from the protein, and they're transient, where the protein may be more permanent.
Right.
But there's a paper like somebody said, this was like really old. I said, oh, it's disappointing. But now we understand what the situation is. And with Tisney, it doesn't matter anyway, because we can you can group the B cells, they will group together, and any particular B cell marker that we know is definitely on the B cell is not necessarily expressed at the gene level. So we understand completely, and we're not even interested in it. It's nice if I was interested in mRNA half-lives, I would study this, but that's
But you can't use it to really validate.
Well, you can't use one gene as you know is on a B cell to validate to pull out the B cells.
Yeah, I mean, because like that was what I was gonna say. If we just need genes, that would definitely pull out just B cells. Like there are probably 4
Right.
that with probably the sum or maybe the log sum
It's
of
like a
all
signature.
yes, would definitely pull out just B cells.
So what are the other 3?
MS4A1.
Can you try that one?
So MS4A1. Yeah, that.
Okay.
And then the 2 CD79s. 79A and B.
Oh,
So this is gonna highlight all of these.
Yeah, so those would highlight all the B cells with CD19, I guess.
yeah, you need a vector or something. You need like a eigenvector. This, or this, and that makes B cells.
Yeah.
But actually, you know what? We're not that interested in this that much because we already know the answer.
Yeah, but you could probably throw out that line at the top.
Yeah.
Right.
In the cluster 9.
Yeah, it looks like this line up here, and then some of the ones down here aren't a part of these.
Right.
Right. Oh yeah. No, we know that to be the case. Yeah, that's
Yeah.
clear. And that's not even the question. It would be nice to get rid of them and do the Disney plot without those having an influence as a weight on the other one because they must be a pretty big weight. But that's for somebody else to worry about why B cells don't always express all the markers they should.
Yeah, so it's almost
We're off the track trying to figure out why non-B cells, why all the B cells don't express all the genes B cells should.
I think it's mostly just cluster 9 and 11 that are.
Right, yeah, it is.
So,
Cluster 9, 11, we will eliminate them from every plot we make, and it'd be nice to eliminate them from the analysis.
So it'd be, then I guess you can't go and redo another embedding without those cells, can you?
That would be nice to do that. That's what we asked the guy.
I guess the way you'd have to do it is just export those cells, go back to the input file, eliminate them, and redo this.
That would be very good to do that. I'd be all in favor of that. Nobody
Yeah.
seems to be able to do that yet.
And it's
No, that should be capable of doing it in like Seurat or something like
Yeah.
that. We should be able to do it.
Can you do that? Because that would be,
can't do it.
and
In the
especially,
program, no,
in-
just do it manually.
and especially if you could do it for the 5 prime, because now you're back on the 3 prime people, right?
Right, so now you want to go.
Because,
5'9".
I mean, it'd be nice for that 1/6, is that one, whatever that, who's me, but anyway, whatever that one is over on the right, I'm interested in that person, but, we want to get a result with the 3 prime, I should say, the 5 prime one, if you go back to that, because let's drill down on that and do some of your magic with the B cell receptor. Is that possible to do? Those 3 5 prime guys. Because I'm telling you, we got to put it in a grant. We want to get a good figure. We want to see what it's going to look like. The grant I'm writing right now, and what we decide, either we'll decide it or imagine it. What the hypothesis is.
Okay. Yeah, we can definitely look and see if we can pull out all that other stuff and just do the analysis on the ones of interest.
This is 5 prime, right?
This is the 5 prime.
And but Chris was saying, see that heat map? You just drag it out. You just and you get that. And he says then you can export with that little camp. Well, actually, the arrow's not there.
That's because this is not the table view. You have to go back to the table view,
Oh, okay.
which is the one right in the lower left. See,
If you go to because the lower left.
those 3 little icons.
Oh,
Yeah,
right, that one.
Right.
That's stable view.
Yeah.
Yeah.
And then that you can export with the arrow in the upper right.
Okay, and then you can then it puts the genes.
Right. And then you can put it in whatever heat map program, or we can do that.
That would be good. And you can do the top 50, 100, or 1,000. So that would be something to do.
But you can also annotate the clusters based on the
Oh, yeah.
person.
Yeah, that would be good too. All right, so what about the what about the B cell receptor?
So in terms of incorporating that data into this, I haven't done it yet. We should be able to, for this, we should be able to import the files into there.
Yeah.
So what I started to do, I was hoping to have it done by now, but it's not quite. I took that spreadsheet of the joined heavy light chain sequences, and I went through, and there's about 2,000 rows of cells that have BCR data with both heavy and light, although some of them were missing heavy and some were missing light. So I just threw those out. There were some that had V gene calls or J gene calls that were concatenated by semicolon, and the V genes were very different. Like it might have been V169 and V323. I eliminated those. So I'm left with about 1,600 cells that have complete receptors. So
Oh,
either
that's
the kappa or lambda.
Yeah.
And so what I'm going to do with
No,
that.
but you said there's some that are different, right? You said there's some are similar, and that's just a missed call, slight missed call, and some are different, and that's something strange.
No, it's
What's
just
this?
normally there's a column that says IGHV column, and most of the time it would say something like IGHV 323.
Okay.
Sometimes it would say IGH 323 semicolon IGH 459.
Oh. I
So
see.
my naive interpretation was that it was an ambiguous call, or it was poor quality. So for the purposes of this exercise, I just eliminated those.
That's good. And it sounds like it's about 10%.
Well, it turns out we started with 2,000, and I narrowed it down to 1,600. And then what I was going to do is I'm going to import it into MATLAB and essentially cluster them. So I'm going to concatenate the heavy V, the heavy J, the light V, the light J, and the CDR3 length of the heavy, and then put everything into a bucket. And if there are more cells, if there are multiple rows in that table per bucket, that means that the CDR3 similarity was not exactly 100%.
Mm-hmm.
And so then I was going to go see if there were, so it calls clonotypes based on 100% CDR3 similarity. I was going to see whether we could broaden the clonotypes by, say, 85%. And then we would have we would have a clonotype that's defined by VJs and lengths. And we have this, I think there was a cell ID in that, I don't recall. But
Yeah.
once we have that data, we can see whether, so it tabulates them. I know, like, it says there's a column that says frequency, and it goes from 1 to 6. But if we can combine rows to make a clonotype on a looser CDR3 similarity,
Right.
then the frequency will go up, maybe even higher.
Yeah, that's right.
And then when we have that, clonotypes that have multiple, if we can go back to the cell date, figure out which cells those came from, we can overlay it on the transcription to see which clusters they show up in.
Right, that's exactly. Now, were you talking about the heavy and light chain, or what are you're talking about the CDR3 region, so I guess you're talking about the heavy chain.
Right,
Or
the
the-
CDR3 is only the heavy chain.
Right.
The CDR3 it's mostly the D gene.
Is there any way to pull the light chain in there?
Right, so basically what I was going to do is you can take, so you have like an example would be, so you have V1.2 J5, that's the heavy chain.
Mm.
Then for the light you have V136, and the heavy is going to be J1. So the combination of those four things, plus the length of the CDR3 nucleotide sequence, which, I don't know, let's say 37, or no, it's going to be 39 bases. So those 5 pieces, you can concatenate those 5 pieces of information together and get a unique ID for that clonotype, assuming 100% similarity. Then all I'm going to do is see if we can group some of their clonotypes into broader clonotypes by allowing some mismatches in the CDR3.
Yeah.
And then we'll get a new clonotype identifier for each of these. And then we can, if we find any that are expanded, we could take that and then find out which cells and which clusters they are, so we could link that to the transcriptomic data. But most of these have frequencies of 1. I mean, so the question is are we going to recover, find bigger clonotypes? And so these are pooled B cells, right? So if they're mostly naive cells, you're not likely to find big clonotypes. So we did we're sort of dealing with this from a different perspective on another project. We're working with somebody, and we're going to do 10x, and the question was that we may only get 400 cells, and the question is, can we see expanded lineages in that? But we're dealing with very specific cell populations, either memory B cells or plasmablasts. And so the question was, can you identify large lineages with just 400 cells? And so what we did was, we have that data, we have some preliminary data for bulk RepSeq, and we have tens of thousands or hundreds of thousands of sequences, and we've identified the clonality plots for those. So Chris and I subsampled that data for samples with varying degrees of clonality, some that had just 2 expanded clones and the rest were small, some where there was a distribution of clones, some that was pretty flat. And we subsampled that to 400 sequences, and then clustered those to see if with just 400 sequences, can we detect clones that may occupy 5 or 10% of the total number of sequences that we sample? And the answer was yes. So then looking at this data, if taking these sequences and then relaxing the CDR3 doesn't really buy you much, and we don't get much, we might maybe go from 6 to 10 sequences. I would guess part of that explanation might be that you don't have sorted cells and they could be overwhelmed by naive cells, because I know that when we do sorts we've done subsets of memory cells, plasmablasts, double negatives, things like that. When we have the naive cells, we get order of magnitude more cells that we sort from that. So I suspect you're not going to see huge clonal expansions unless you filter out the naive cells or do some kind of sort before.
Well, we did do a sort we got the data on transitional, plasmablasts.
Right, so that one you have a hope of finding.
Right. So maybe you could take a look at that.
So I was looking at 165, which is just B cells, right?
Right, the
Okay.
total and definitely more naive, mostly naive.
For
Yeah.
165, not 165. For 177, there is naive, like the Tcell 1A. I mean, I'd probably say for 177, it's about half and half. Half naive and half memory.
Okay.
And then for 166, it's probably mostly naive. But then for 165, it's about a third naive, a third the F05, and then like a third memory.
So is that T-bit high?
Yes.
T-bit high memory.
T-bit, yeah, so T-bit high, that would be like about
About a
like
third
a
or
third.
fourth.
Yes.
Maybe because you restricted.
So you have a third naive, a third T-bit high memory,
And then like just memory.
other memory.
Yes.
T-bit negative memory.
Yeah.
But that you've got the data. That's 165. That's when
That's
you
the one I was looking at. Right.
Right.
So,
Yeah.
based on this other data I was just describing was also sorted into T-bit high and or FCRL5 positive negative memory too. And that was 7 and 14 days after vaccine, and so they were very expanded.
Oh yeah, that was.
So there's no intervention in this.
No.
So, okay.
So you don't even know what drug they're on, do you?
Right, that's right. But
So-
similarly, immunosuppressant, but transdermic wise, we did notice a big difference between 165 and the other two. So 165 has autoantibody, liver disease, and she has a lot of the type 1 signature gene, whereas the other two really, in a way, very quiescent. Yeah.
But the one.
If these sorts are very different from person to person, that's going to give you totally different. So you can't really analyze these together.
Right, so can we
Because-
zoom into, for example, just naive, and looking at just the naive population, which is IgG positive?
Well, if you look at the naive population, you're likely to only get one clonotype. You know, your maximum clonotype frequency is going to be 1 or 2 or something like that, because there's a really vast repertoire and you're only sampling 1,000 cells.
I wonder if there's a way to relax things, you know, like stage-wise relax. Because, okay, the D and then relax it and you get the, what I'm trying to think about is going back to whatever the germline gene was. And if you relax D, you might get that because, you know, you relax to the point where all the ones that selected that D, and then you relax J to a point and relax, and then I guess V will be, you can call V pretty clearly, right? I don't think
Yeah, I
anybody
mean, that's a binary. That's really not,
right.
you know, they just assign a V gene to it. It's not,
Right, it's this one or that one.
right.
You know, but I mean, if you don't get clonal expansion, it's supposed D is if you relax D to a point where, you know, here's the thing. I think it's V, D, J, and then they go on, and then from there they expand,
And
you know,
mutate.
drive, and they semantically mutate and so forth and so forth. You know, so if we knew what just call the VDJ that was original at the germline. And you know that would mean relaxing D quite a bit, so you get the germline one, like we discussed before. There's only 6 of them, and then what did they do? So this guy started out in the germline like this, and then what's the end result?
Right, but they're not going to change. It's the same V gene that gets propagated throughout the tree,
Right,
so it's just mutated,
right,
but it doesn't. The gene doesn't change. So.
exactly. So what I'm saying is relax it so much that, let's say we want to look at all each, you know, the V regions, just call that, and which of those are even expanded. I mean, maybe they use a selected a different DJ.
Well, but those are going to be, then you're going to be mixing several genuinely different clonotypes into one cluster.
No, I know, I realize that. I mean, I'm just saying, how much do you have to relax it to get to the question? If you, let's say,
If you do that, that's going to suggest that then there's a difference in gene usage. That more of the cells are expressing a particular V gene. So if you don't, let's see.
no, I think I understand the process. The point is that, you know, if you restrict it like they did, and everything has to be absolutely identical, then there's no clonal expansion.
Well, it could be if it's the exact CDR3, but because
Yeah.
if
Well, it
they're
occurred
mutating-
6 times or something like that. But, you know.
Right, but you're losing the information because the CDR3 ought to be highly mutated if there's some kind of response.
Yeah.
But if they're naive cells, you're not going to see that.
Well, and we do, and getting back to the naive versus, you know, plasmablast question, we do have sort of plasmablast. So that would, I think you got that's in your, that's all in the box.
We have to.
But can I just make this one point? Here's the thing, whatever anybody did before is fine, but what I want to do is do something a little different, maybe, it sounds like. You know, go step by step, because I'm in the discovery mode. I don't assume anybody knows anything, you know, because I figure most people haven't worked this out very well. And I figured that, here's the situation. Based on, let's say the V region counts all by itself, the D and J count, but I don't focus on that. But the reason why, and why historically people focus on that, because that's what they had to focus on. But I'll just say, okay, fine, you get a B cell has V range of this V, and let's throw in the light chain these 2 V's. You know, how much is there clonality based on that? Yes, no, okay, fine, but you know there might be, but then okay, but you forgot that you have a joining in the D in a joining region, and the same they've got a J over here with the light chain. So add that in, and how is there clonality?
Right, but so that it sounds like clonality is really going to be a function of the joining of the heavy and the light chains in the particular V and J genes. But
Why do you say that?
Because if you have 2 sequences that they might have the same V heavy and V light, but different J's, they're going to be different.
What if J doesn't matter too much? What if it's a low affinity antigen?
So you're not really asking about clonotypes. You're asking about things that might be reactive to the same thing.
Yes,
I
exactly.
see what you're saying. Okay.
See, I don't care what everybody did before. So,
okay.
The CDR3 region there's you know, mutations, high semantic mutation. That's really important. It's long.
Right. So it's a question of gene usage then.
Right.
You're
Why
asking-
do they come up with this thing? Because they twist and squeeze the data, and they have diversity. And I know what Harry Schrader does. They have diversity region data. That's it. We're going to make something out of it because we're not going to study this do all this sequencing and come up with nothing. So they squeeze that. But there, meanwhile, we got new data that's never been seen before. We got V heavy and V light. So ask a different question. Does that do
Got it.
anything?
So you're asking, so start from the broad, cast the widest net and say,
exactly.
is there a bias in V gene usage? Then is there a bias in both V heavy
Pairs,
plus
yeah.
V
Right,
light?
yes, this is
Then
very
add the J's in.
Forget all the stuff that anybody thinks they know, because they're probably wrong. I think it's a
So-
low affinity interaction. The D and the J may not matter very much. The V's may determine a lot of it. It's not, you know.
Right, so the selected antibodies might be preferential for particular V genes that would show up.
Exactly. Yes. And the thing
So-
is we know, because we did it. We looked at the antigen binding of antibodies, and they are generally low affinity. It's a funny thing, because this somatic height mutation focuses, because this came up many years ago, but you looked at, I mean, you have limited look, but the thing is all this D and J, finally the antibody recognizes everything. You know, because we did it. We took a given antibody, an autoreactive antibody, and they have this broad spectrum of reactivity. And you say, well, how can it focus on anything? Yeah, I know, maybe there's a arginine, modification. Remember this? I remember giving this talk 10 or 15 years ago when we first discovered AID was elevated, and there's still debate about that, but AID is elevated. And then somebody says, I remember explaining to somebody, here's a single antibody, and put it on a Western blot, bound to 10 different things. And I said, so here it is, highly somatically mutated, and it binds to everything. And then somebody in the audience jumped up and says, how can you have something focused down, 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 and then it binds everything? So it still doesn't make sense, but I'm just saying that whatever you hear from anybody else in autoimmunity, they're probably not 100% right. So we're just willing to look at it again,
Right.
and especially considering the light chain.
So we can, that's easy enough to do. The question is then, what is your reference going to be? So suppose we have one sample, maybe a lupus patient that's undergoing a flare, and we characterize that. How are you going to know whether you just look at the V's only or look at the entire clonotype? How are you going to know that's abnormal?
Well,
or different?
my approach to these things is look at everything. Yeah, I know that might be asking a lot.
No,
Look at
you can do
V's.
that, but that's one individual, and so that individual has some characteristic, some bias in V gene usage or some clonality profile or something like that. That tells you for that individual with
The thing
these
is,
conditions.
Oh,
So are you
yeah.
gonna?
in the same individual, we do find this patient has very high neural signature, but within the same individual, that their B cell, which don't have that signature.
Yeah,
So
well,
then we
okay, to
compare
answer the
it.
question, that's where the transcriptomics come in. Because again, we have a model
Mm.
where the B cells that are autoreactive have a transcriptome that favors their survival.
Right, so
And
you can then look at the V gene usage for the different transcriptomically defined
Right.
B cell.
If we see,
populations.
here's actually the thing is, we think we know the transcriptomic signature of an autoreactive B cell, or we have two different signatures now. And if we take that, I don't know how exactly how to do it, but we take those B cells with that signature and select them, maybe there's 500, and look, is there any clonality by any way in those versus the ones that don't have that signature? Is there a difference? That's really what, and I don't care what the difference is, if it's heavy light chain. V.
Actually, so what are the signature genes that define? Can we just look at those now for the?
Yeah.
Which are the ones
that?
that-
I think ISG 15, ISG 20.
ISG 15.
Yeah, so there are few, and MX1, those are the classical. type
If you
ones.
look at-
If you're on
Right here.
signatures.
Right.
What am I looking for?
ISG 15.
ISG 15.
Oh, he can't hear, okay. He's can't do
Right,
that.
right here. Right,
That's everything.
right.
And then what other?
And then ISG, so for 165, ISG 15, ISG 20, and then IFIT M1, 20.
Sorry, I think because she's a little bit further from the microphone, I can't.
Oh.
Can you-
20, 20,
20?
yeah, 20, you have it there.
Okay.
Yeah, we can get this over closer. This is the microphone.
Right.
That's the camera. Oh, it's also the mic.
I think the mic's in
Yeah.
this. Hello, can you
Yeah,
hear?
yeah,
I think there's a mic.
And
Yeah.
then,
Imagine being
and then
there.
IFIT M1.
M1.
Yeah, IFIT, not ISG, IFIT M1.
iFit1?
Right.
Oh, iFit-M1, right,
Right.
yeah.
Right.
Yeah, actually,
So we can,
hey, Chris,
Yep.
is it possible to export the coordinates of a CC plot?
That's like.
Not
What
the
do you mean by
picture?